to the third day of the one-week international seminar and model presentation on stepping down from research to the world of reality, an outcome-based approach. Our speaker for today, Dr. Shagnik Chatterjee, is currently working as a senior research scientist for Ferring Pharmaceutical, Copenhagen. His contribution to this biotechnology field includes as being senior research investigator at Biocon, Bristol Myers Squibb Research Center, and working as a research associate at AstraZeneca India Private Limited. His education started from acquiring an MS in pharmaceutical in NIPR Mohali, India, pre-doc in drug delivery and disposition in KU Leuven, and ultimately PhD in drug in the same from KU Leuven. He was also entrusted with responsibilities like scientific advisors to the editor in Journal of Pharmaceutical Sciences and executive guest editor of Current Drug Metabolism, and many more. Sir, the screen is all yours.
Thank you for the kind introduction. You do not need to call me sir because I'm yet to be knighthooded by the Queen. So my name is Shagnik, and I will try. I've not done this with Google Meet ever. I have done this with mostly Zoom or MS Teams. So just let me know if this is working or not. Can everyone see my screen?
Yes, sir, you're presenting.
Yep,
yes,
thank you. And please interrupt me during my presentation for any questions. I'll be very happy. Rather, if I do not get any questions, I'll get demotivated in speaking for like 1 hour only myself. So I'll be very happy if someone asks a question, basically. So and I saw one model in the brochure that talks about transmembrane pumps. So today I'll be giving you basics about transporters. Transporters are nothing but transmembrane pumps. So basically, from one side of the membrane, it transports compounds from one side of the membrane to the other side of the membrane. That's what transporters do. So first is whatever I know so far is because of a lot of people who has taught me either formally or informally via different discussions, and whatever I know is because of them, thanks to them. So these are all the people that has provided me the knowledge. I am not going to discuss about either the movie transporter or either Uber or Ola or anything. So it is a drug transporter. It's compound transporters. So what are the things that I will try to cover, and like different transport processes? What are the different types of transport processes that goes on in a cellular environment? Localization of the transporters, the effect or impact of transporters in drug interactions, toxicity, clearance, and pharmacokinetics. And finally, some of the transporters are now used as a drug target as well. Again, please interrupt me for any question at any time. I'll be very happy to answer the questions. So transporters are present in all membrane barriers. Whenever there is a cellular membrane that differentiate two different environments. Example, what I mean by two different environments. So in our intestine, there is that intestinal, what I say, the trunk, which is separate from body's inside environment. Whatever we eat goes in basically that intestine, and the important molecules or the nutrients gets absorbed.
Excuse
from
me,
the
sir.
intestine. Yeah, sure.
Can you please switch it to the full screen mode? Because the whole window is being shown.
Can you not see the whole window? Because I can see the whole window.
Yes, I can see the whole window, but the PPT is not being presented as such. It's looking a bit smaller.
Okay. Hmm. Because I'm present doing it in slideshow mode only. I'm not doing it in the
Okay.
smaller mode. Can you see it now?
No, sir. Can you present it?
Nope.
It's not presented.
Screen share is
First, you
off,
should
sir.
share the screen, sir.
I am sharing the screen, but I don't know. Wait, I will try it again. Entire screen. Damn, your entire screen. So you cannot share the entire screen. Why? Share.
So now we can see your screen.
Now you can see my
Now, sir.
screen.
Yes, now it's
Can you
being
see?
presented. Yes, sir. Yes, sir.
Okay.
Now
Okay.
you have changed the
Okay.
slides, right? Yes, sir.
Yeah. So, yeah, transporters are present in all membrane barriers that differentiate one compartment of that differentiate between two different compartments. Now, when I say compartments, it can be external environment from body's internal environment. So, the intestine is external environment. When the compounds gets absorbed into the portal vein via the mesenteric veins, these are internal environment. And similarly, another two excretion mechanisms or excretory mechanisms of body are either via bile or via urine. So in that separation place, in the places that separate bile from blood or urine from blood, transporters are pretty important. In general, every membrane contains transporters. We just don't know all of the transporters yet. It is estimated that there are about 500 to 600 transporters present, and we know out of the clinical impact of around 20 to 25 of them are shown. So rest of them are yet to be discovered, or the clinical impact is yet to be shown. So there are different types of transport processes. There is called active transport and passive transport. So active transport is when the compound is transported against the concentration gradient. So when I say against the concentration gradient means that compound may get transported from a region of lower concentration to a region of higher concentration via a transporter, and that requires energy. So active transport is always associated with some form of energy. Some of the energy source we already know, it has already been discovered. Some of the sources are yet to be discovered, like ATP is one of such energy source used by ATP binding cassette transporters, which is called, these are called ABC transporters. And there are other type of transport which is called passive transport or passive diffusion, which is basically stops when the concentration, the free concentration in both sides of the membrane becomes equal. That's passive diffusion. That usually happens from a region of high concentration to a region of low concentration. And yeah, active transport is against the more often than not against the concentration gradient. It can happen towards the concentration gradient as well. There are exceptions, or there are examples of that process, but mostly it is against the concentration gradient. Now, the functions of transporters are basically gatekeepers. So if you think of a gate, and if you are the gatekeeper, you let one person go inside the gate from one part of the house to the other part of the house, while you do not let few people, or the people that can cause nuisance, to go inside your house. So that gatekeeper's job is basically the transporters. They supply nutrients like glucose transporters, SGLTs, maintain cell homeostasis, like this is called BSEP, bile salt export pump. So it effluxes out bile salts or bile acids from body or liver to the bile. So once the compound is in bile, it's going to get excreted. So this is what BSEP do, and there are numerous other transporters, and there are lots of other examples. Sometimes they play very important part in drug absorption, disposition, or elimination. The major evolutionary mechanism why we have transporter is basically to I mean, the most important function is to remove the toxins. Now, what are toxins? Anything that is not within your body. That's basically body thinks that as toxins or xenobiotics. So the evolution wise, the transporters are made to remove the compounds from your body so that it does not take inside any foreign compounds. Now, transporters is not the impact of transporters. It's not a new thing. In Second World War, during Second World War, one of the most important antibiotic was penicillin, but penicillin was getting removed from the body very quickly. So what they found out that if they give a compound called para-aminohippuric acid, the retention time of penicillin inside your body is increasing. Therefore, the function or the activity of penicillin also increases. At that time, people did not know that there is a very important transporter that plays a part in this process, and that's called OATs, like organic anion transporter, that's present in the proximal tubule cell lines in proximal tubule cells of kidney. So penicillin is a substrate of OATs. So what OAT do? OAT effluxes penicillin from the body into the urine. What paraminohippuric acid do? Paraminohippuric acid inhibits OAT. So penicillin cannot be removed via urine anymore. It stays in body for longer time, and thus the activity or the function increases. Any questions? Yeah, please interrupt anytime. As I was discussing previously, that there are a lot of transporters, 400, and nowadays 500 transporters have been identified, but only 20 to 25 of them we know that there are clinical significance of those transporters. Now, how do we know how this clinical significance come into light? When we see drug-drug interactions, when we see some unexpected behavior of any compound, unexpected in terms of pharmacokinetics, or it can be in terms of toxicity as well. In any specific population, and then possibly after some time we realize that there is a transporter gene that is either absent or hyperactive in that specific population, and that's leading to that specific phenomena. So this is how the impact of transporters are usually discovered or comes into light. Now, as I was saying, that transporters are present everywhere. And as the name suggests, it transports from one side of the membrane to the other side. Now, if you look at the intestine or the intestinal epithelium cells, there's the cell line border that protects your intestine. So as you can see, the lumen of intestine, the compounds come from the lumen of intestine, then they gets absorbed into the enterocytes or the intestinal epithelia, and then they gets into the blood. So it's these cells work as a barrier between intestinal environment and your body's inside environment. There are lots of different transporters present here. Some some of the names we know commonly, like PGP, P glycoprotein MDR1, that's in purple color, or yeah, I don't know what color is that. Red color, you can say. So PGP, BCRP, if you look at the arrow direction, these are from inside the cell or the inside the intestinal epithelia into the lumen intestine. These are called efflux transporters. The efflux is compound from inside the cell to the lumen. There are other type of transporters like ASBT, MCT1. ASBT is apical sodium bile acid transporter. So it absorbs the bile acids from lumen of intestine into the intestinal epithelia. Then there are other transporters in the other side of the membrane, which is called basolateral membrane or the blood side. It's called OST alpha beta MRP3. Again, these are all the names of the transporters that's been there in different articles, and you can easily get. But what you want should understand that transporters basically transports, and by transporting they regulate the concentration of the compound inside your body. Either they can throw out compound from your body to lumen, or they can take up compound as well, like OATP2B1, like ASBT. These are uptake transporters. Similarly, in kidney proximal tubules, the similar thing happens. It's also a barrier between blood and urine. So you can see there are a lot of transporters that the arrow is facing inside the cell. So compounds from blood go into the proximal tubules and finally get eliminated into the urine. These are OAT1, OCT2. So OAT1 is one of the transporters that absorbs penicillin. And similarly, on the urinary side, there are efflux transporters like MET1, MET2K, MDR1, PGP again. So one transporter can present in different places. It's not that one transporter has to be present in one specific tissue. The tissue specificity is sometimes there, sometimes not there. Like OCT1 is a transporter that's present only specifically in liver, while OCT2 is present in kidney. So it's more into the nitty gritty. But basically, what you have to remember is that any small molecule, specifically if they are more polar in nature, they will require transporters to cross a membrane domain, and thus the concentration of those compounds inside the body will be governed by transporter expression, transporter function, and everything. Now, if you come to the liver, which is the major elimination organ in your body, this is a simplistic picture of a hepatocytes, the major cell type of liver. So there are two. Try to see. Yeah, so this side of the cell membrane or the hepatocytes membrane faces the blood, while this side is the bile. So this is the basolateral side or the blood side, and the bile side or the canalicular side. There are different types of transporters present in the blood side, and there are different types of transporters present in the bile side. Again, some of the transporters like OATP1B1, 1B3, they absorb, they take some compound inside the hepatocytes from blood, while there are other transporters like BSEP, MRP2, MDR3, they effluxes from the blood into the bile, while there are other transporters like MRP3, MRP4, West alpha beta, they effluxes compounds from hepatocytes into the blood. Not the same compounds as substrates of every transporters. There are very specific transporters do have very specific substrate requirements. So example, OATP 1B1, 1B3, they transports mostly the statins, a sort of anionic compounds, while PGP, BCRP, mostly the cationic compounds are substrate of those transporters. You can read more about this part in lot of literature articles coming from ITC, International Transporter Consortium. They publishes or updates the transporter knowledge every 4 year. Just last month, their last or the most recent version of it came in 2022. You can refer to that as well. Now, one of the major impact of transporters is in drug interactions, which is called DDI, drug-drug interactions. Even this is a simplistic depiction of a transporter mediated interaction. So if a compound if a compound gets into the liver via OATP, and if another compound inhibits OATP, then that compound cannot get into liver. So if the liver is the target, then the efficacy will decrease, or if it affects another organ of your body, then the concentration or the systemic concentration of the compound will increase, and that will lead to toxicity. And there are lots of compounds that have been withdrawn from the market because of this. Example, trogridazone. Intestinal transporters. So some of the drug interactions that are more famous in the intestinal domain or the intestine. So these are the enterocytes. That's your portal blood, and this is the gut lumen or intestine. There are different transporters like OATP, PEPT that absorbs. That's in the absorbatory direction, or that takes up compounds from lumen into the enterocytes, while there are other transporters like PGP, BCR, PMRP2 that throws out, it fluxes out compounds from enterocytes into the gut lumen, while there are other transporters that takes up compound from enterocyte into the portal blood and then get into the liver. Some of the most famous drug interaction cases are like quinidine digoxin. So digoxin is a substrate of PGP. It usually digoxin what it gets effluxed out from the enterocyte via PGP into the gut lumen. If a compound inhibits this efflux, then the concentration of digoxin will increase in the enterocyte and ultimately will lead to 1.7-fold increase in AUC of digoxin. AUC is area under the curve or systemic concentration, you can say, of digoxin. Now, 1.7-fold increase is a very serious thing that can lead to death sometimes also, depending on the therapeutic window of the drug. Digoxin is a low therapeutic window drug. Similarly, there are other examples like topotecan and then rifampin digoxin. This is ilacridar GF12098. So again, as you see, if there is an inhibition, there is an increase of concentration of the substrate. While if there is an induction of PGP, so the PGP expression increases in the intestine, then there is a decrease in the plasma concentration of digoxin. Hepatic transporters, do we know anything about hepatic transporters? Yes, we know a lot. So some of those compounds are substrates of, like the statins, these are substrates of OATP, 1B1, 1B3. So a compound is absorbed from sinusoidal blood via OATP into the hepatocytes. Then in the hepatocytes, they get effluxed out into the bile. If another compound inhibits OATP, then the compound cannot get into the liver and the systemic concentration will increase. This is what happens when cyclosporine is an inhibitor of OATP and atorvastatin is a substrate. So atorvastatin concentration increase in case of a OATP inhibition. And similar thing happens for other OATP substrate as well. This is one of the seminal, most important studies done initially by Professor Sugiyama's group in 2011, I think. Yeah. So a compound can have different mechanisms of elimination via cytochrome P450 enzymes or via transporters. So if there are different elimination processes, how do we know the relative contribution of one process over another? We can determine the rates of those processes, and the slowest step becomes the rate determining step. That's one way to do it. Another way, inhibit one process at a time and see what is the impact of the systemic concentration of the drug. So here, I think they have taken atorvastatin again, which is a substrate of CYP3A4, and also OATP1B1, 1B3. And if you give a CYP3A inhibitor, itraconazole, the AUC does not change by a lot, like twofold. While if you give rifampicin, which is a OATP inhibitor, the AUC changes by 22-fold. AUC is a measure of plasma concentration or the systemic concentration. Similar type of things happen in case of renal transporters as well. So there are compounds that are substrates of renal transporters, so OAT 1, 2, 3, OCT 2, OATP 4C1. There are lots of other transporters, uptake transporters are there. And if you have a compound which is an inhibitor of those transporters, it will change the AUC, and that will lead to change in toxicity or exposure or efficacy of the drug. In some cases, like this metformin, you see the impact of transporters only in case of tissue concentration and not in systemic concentration. So metformin is a compound which is a substrate of liver OCT1, also kidney OCT2. So if you inhibit liver OCT1, it still has kidney OCT2 to take care of the excess load of transporter or of the compound in the body. And that's why you do not see a change in systemic concentration if you inhibit OCT1. However, the liver concentration of metformin changes. And why it's important? Because the target is in liver. So the efficacy or the diabetic efficacy, reduction in blood sugar also decreases. Transporters can lead to toxicity as well, similarly. Well, any questions so far? Possibly no. So I'm not doing a good job in explaining. Okay. So transporters can lead to toxicity. Transporter inhibition can lead to toxicity as well. So say this pink color dot is a bile acid. Suppose example. So bile acids are taken up inside the liver, then they go into bile. If a compound inhibits this excretion into the bile, the concentration inside the liver will increase. The volume of liver cells, hepatocytes, is way low compared in the microliter, picoliter range compared to blood volume. So the concentration of the compound will increase a lot, and that will cause toxicity. One of the most famous story about toxicity of mediated by transporter is by serivastatin and gemfibrozil. So serivastatin was launched by Bayer. It had 500 cases of rhabdomyolysis, including 100 death. Now, out of those 500 cases, half of the patients were on gemfibrozil. So why it happens? When they gave gemfibrozil compared to placebo, the serivastatin concentration increased a lot, like 5-fold or something, 5.6-fold compared to placebo. But they knew that it's a substrate of CYPs. So what happens if there is a CYP inhibitor? The concentration does not change. Only by 15 to 20% increase of AUC with CYP inhibitors, itraconazole and erythromycin. But concentration increases up to 5.6-fold with gemfibrozil. So serivastatin is a substrate of OATPs, and gemfibrozil is a inhibitor of OATP, and concentration increases, and that leads to systemic toxicity of serivastatin. There are other statins where there are some pharmacogenomics evidences have shown that some of the mutations in OATPs increases the exposure and risk of myopathy for simvastatin as well. Similar drug interactions has been noted for other compounds like donepezil and cilostazol. Donepezil is a substrate of PGP and BCRP, while cilostazol is an inhibitor. Now, donepezil accumulates in the heart and brain because cilostazol inhibits BCRP, and that's why donepezil is toxic in presence of cilostazol. But if it is given alone, donepezil is removed from the heart and the brain by the transporters BCRP. Now there is a phenomenon called drug-induced cholestasis. That's also another type of liver injury or toxicity that's mediated by some of the drugs, compounds. Drugs means medicines here. So this yellow dot is a bile acids. The life story of bile acids is such that bile acids are synthesized in the liver. They accumulate after synthesis. They are excreted into the bile duct, and they accumulate in the gallbladder. When we have a meal, the gallbladder is contracted, and the bile acids get into the intestinal lumen. The functions of bile acid is to basically help in absorption in fat soluble vitamins, and they get recirculated via the portal vein. This is usually they call it as a enterohepatic circulation of bile acids. But there are some compounds, they block the efflux of bile acids into the liver, into the bile, and it leads to accumulation of bile acids in the liver. Bile acids are toxic, can lead it can cause direct plasma membrane damage, which is usually not the case. However, it can definitely cause inflammation at the pathophysiologically relevant concentrations. And also, there are some evidences of apoptosis by some of the bile acids. This is another way of representing the same phenomena. So here the bile acids are depicted as blue dots, blue circles. Bile acids are taken inside by NTCP, sodium dependent taurocolate co-transporting polypeptide, NTCP. And if there are and they are thrown inside the bile canaliculi via BSEP, bile salt export pump, another transporter. And if a compound inhibits BSEP, then it will lead to increase in bile acid concentration in the liver, and it will cause damage of the liver. One such very famous case study is trogidazone. Trogidazone was initiated or taken into market by Pfizer in 1999, and it has to be withdrawn by 2001 because of drug-induced cholestasis. Any questions so far? How much time do I have?
It is 4:34.
Hello? How
Sir,
much
it
time
is
more?
4:34.
Okay, how much more I
Around
have?
half an hour. Around
Half
half
an
an
hour.
hour.
But you have to be engaging. Have you understood anything, or I'm really not doing a good job here? Okay, I will continue the rest of my presentation. Transporters can impact clearance as well. I do not know whether you guys know about clearance or not, but yeah, without knowing, it's very difficult to explain clearance. So clearance is basically a parameter that calculates or that suggests the excretion rate of compound from your body. That that's what you can understand from clearance. So the faster the clearance, the faster the compound will get out of your body, and lower will be the efficacy or the exposure. So if a compound is taken inside the hepatocytes by OATPs, it can be metabolized inside the hepatocytes by cytochrome P450s to get into the bile. But there can be two different rates of these two processes, and finally the slowest step will determine the rate of the overall pharmacokinetics or overall PK. The rate limiting steps in drug clearance, one of the initial papers came out about it is from Professor Brian Houston's group in Manchester University. So they showed that transporters actually can govern clearance of a compound for the first time. So this is what they did. Basically, they measured the clearance of compounds in different systems like microsomes. What are microsomes? Microsomes are basically endoplasmic reticulum containing vesicles that has the cytochrome P450 enzymes, while hepatocytes are intact cells, whole cells, and you can measure the clearance of a compound in different systems like hepatocytes or microsomes. And if you look at different numbers, you can see the final hepatocytes clearance is similar to the hepatocytes permeability, which is basically that measures the rate of uptake of compound into the hepatocytes compared to microsomal clearance, which is mediated by cytochrome P450s. And by plotting a general Michaelis Menten curve type of thing, they can show that yes, indeed, uptake is the slowest step or the rate determining step. Now, how to integrate transporter data into clearance and how to calculate the clearance, and basically how to calculate final clearance and human dose is something very important. So this is a basic picture of a well-starred equation. Clearance, hepatocyte clearance, CLH, quantifies the loss of drugs during its passage through the liver. This is the equation of a well-starred model. This Q is the liver blood flow, AB is the fraction unbound, clint is clearance, the intrinsic clearance coming from different systems. And that's how hepatic clearance is calculated. But extended clearance is something else. Extended clearance takes into account uptake and passive diffusion and metabolism and biliary clearance, but it is not a one-to-one equation. It's a pretty complex one. There can be, based on this extended clearance equation, it can be either rapid equilibrium or uptake determined. In case of rapid equilibrium, the passive diffusion of compounds are pretty high. These are lipophilic compounds, while uptake determined are usually polar compounds. The passive diffusion is pretty low. Yeah, that's it. So the take-home points are uptake
Sir,
can determine.
I'm
Yeah.
sorry to interrupt. There's a question in the chat box from Ankon Das. How can we
Mhm.
make sure the drug will go to hepatocytes and not any other cell?
We cannot make sure. The
Okay.
simple answer is we cannot make sure. What we have to do is to limit the entry of the drug to hepatocytes and not to anywhere else. But remember one thing, drugs or any medicines will travel through blood. So wherever blood go, compound will travel through those places. Small
Okay,
molecules,
sir.
I'm saying small molecules. I'm not saying any other mode of delivery here. So it is very difficult. It's not physiologically possible to restrict if you are using a small molecule via a oral route. But there are other means when you change the dosage form, that there is a very specific receptor or very specific protein that is expressed in only one organ, and your compound will specifically bind to that organ. That's more of like MABs. the dosage form become different. You cannot you cannot have MABs as an oral dosage form. Monoclonal antibodies, MABs means. I hope I have partially given the answer. The total answer will require a complete 1 hour class with just one slide, not these 40 slides. Yep, so the compounds can be classified in different classes based on their passive permeability, their metabolism, and their uptake rates. So the take-home points are: uptake can determine the rate of hepatic clearance if it is sufficiently low. For compounds with high passive diffusion, metabolic clearance possibly be the best predictor of overall clearance. Thank you.
Thank you so much, sir, for giving us this insight into transporters, gates, and clearance. Thank you so much for gracing this seminar. It would be very helpful if you could give us something related to this PPT, or even this PPT, if it's possible. It would be
Give us
helpful.
something means papers or?
Yes,
Yes, I
sir.
will send you some of the
Any material
publications that you
related
guys
to
can
this.
look around.
Okay, sir.
Yeah,
Okay,
definitely.
sir. That would be
Okay, sure.
helpful. And it would also be helpful if you could be active on your email so that any of us can reach you if we have any problems regarding to this.
Definitely, you can reach me in Facebook, in LinkedIn, anywhere if there is any questions
Okay,
regarding
sir.
any of these things.
Okay, sir.
Okay.
Thank you, sir. And if anybody, okay, sir. Thank you,
Yeah.
sir.
If
So
in
we would like to wrap up this session. And if anybody has any
Thank you.
questions, please ask.
Yeah, if no questions, then possibly we can wrap it up and have a
Yes.
very good rest of the season.
Yes, sir. Thank you so much, sir.
Thank you.